7, 6, 5, 4, 3, 2, 1.
Welcome to SIYSS 2023 and the fourth session for today. We will be witnessing and take part of 4 presentations from young, brilliant minds sharing their research today. And to kick off this seminar, I need some help, so I want you to give a big round of applause for Elin Palm. Hi Elin, how are you?
I'm doing great. It's been such an amazing day here at SIYSS.
I think so too. For those in the audience who don't know about who the participants are, can you tell us about that?
Yes, so this year at SIYSS we have 18 participants here live in Stockholm. They come from 14 different nations. And they're all in the age of 18 to 25 and have won or gotten their spot here through some sort of international competition or were been sent here by their university due to the high quality of the science they are conducting.
So if you're watching this on the live stream or live here physically in this room and you get inspired to also do something like this, how could a journey look like to end up on a stage like this?
So I assume most of the people listening in here are Swedish high school students. Then I recommend you that during your diploma project, you pursue a very scientific and researchy diploma project. And if you compete with that project in utställningen that Orga Forskare are also organizing, the exhibition, then you might be one of the lucky winners who get to compete with your project on one of the international competitions, like the European competitions for young scientists or the International Science and Engineering Fair. And both of these competitions have SIYSS as one of the rewards that the participant can win, and that's how we got some of the participants here today as well. You can also attend one of our associated universities like Lund University or Cambridge. We're also sending participants to SIYSS based by choosing or picking a certain young scientist that they think have conducted excellent research at their school.
And final question. This event, this seminar, this week, that SIYSS is actually a week, it's just not just today, it's organized by the Federation of Young Scientists, Unga Forskare in Swedish. Can you tell us a little bit about this organization?
The Federation of Young Scientists is one of Sweden's biggest youth organization, and its goal or mission is to inspire youth into science. And we want science to be as a clear and optional spare time activity or hobby as any other hobbies there are, like football or gaming or Netflix or whatever it is that you do on your spare time, enjoying lots of TikToks movies, I don't know. So we want science to be as good of an option as all other options. And we do this in multiple ways by organizing SIYSS, the day we're all attending here today. But we're also having utställningar where you can compete with your high school diploma project and certain other events happening throughout the year to inspire to science. Also a really cool thing that I think we're organizing are the summer research schools where you might get some tips and ideas to become the scientist that can potentially be part of SIYSS one day.
Great. Give it up for Elin Palm. I will soon present the first presenter, but I want to introduce first Arman. Please join me. A big round of applause. Arman, you're kind of my sidekick here today, but you're giving all the good questions to the presenters, right?
Yeah, exactly. So you will all have seen a Menti link, and you can use that to submit questions, and I will basically be looking through those questions and finding interesting ones to ask the participants. I would also like to make a quick shout out to a summer research camp, similar to what Elin mentioned, that is done by Unga Forskare called Raise for Excellence. I'm an organizer for next year, so if any of you are second-year students in high school, do apply to that. You'll get a chance to research in a real lab and increase your chances of getting into SIYSS.
Amazing. Thank you, Arman. So apply to Race for Excellence and use the Menti link to ask questions for our participants today. Okay. This first project confirmed the possibility of using metal organic frameworks as long-term antioxidants. From Korea, give it up for Woojin Park.
Thank you. Oh, okay. So hello, I'm Woojin Park, and I'm currently a freshman at KAIST coming from South Korea. And today I'm here to introduce you how metal organic frameworks can be used as long-term antioxidants. So to briefly explain the motivation of my research, I first want to bring up the concept of free radicals. Free radicals are chemical species that have high reactivity, so when they come in contact with cells, they induce damage such as DNA damage or possibly even lead to cancer. Antioxidants, on the other hand, are a different breed of chemical species that could inhibit free radicals. These antioxidant effects are required for optimal cellular health. However, not enough consumption of fruits and vegetables that are rich in antioxidants are occurring these days. Moreover, one thing about antioxidants is that they have a relatively short circulation time, which means that once you ingest these types of antioxidants, they get expelled in a relatively short period of time. Therefore, we sought the need for long-term antioxidants that could circulate in a maximum amount of time inside our body. We tried to solve this problem with a concept called metal organic frameworks. Now, this chemical structure on the right might seem complex, but you can imagine it being just like this. So picture a cube inside your head. A cube can be comprised of two components, an edge and a vertex component. Now bring that image up into the screen, where the metal center corresponds to the vertex, and the organic linker corresponding to the edge. So the vertex and edges come together in a self-assembled manner to synthesize these kind of intricate structures. Prior research has elucidated two methods to utilize these metal organic frameworks as antioxidants. First, we use the ligands, one of the subunits themselves, as antioxidants, or they could encapsulate the antioxidants inside the empty spaces, the pores of the moth. We decided to follow the first path, assuming that once these moths are ingested and circulating throughout our body, their chemical or physical disintegration could lead to continuous release of antioxidants, therefore enhancing the circulation time. First off, we chose a total of 3 candidates in which we assumed when their degradation happens, their ligands would have antioxidant properties, which are SC101, FETHQ, and CuC81, having the reactants as shown in the screen. These moths are synthesized according to prior research methods and led to successful characterization, as you can see in the scanning electron microscope image on the right. Further, structural determination analysis was conducted using X-rays, IR, and other thermogravimetric analysis methods. So once we have synthesized these moths, we set up a total of 3 criteria to evaluate their performance as being used for long-term antioxidants, in which the first would be obviously biocompatibility. So neural stem cells were mixed with certain concentration of moth and their precursors and cultivated for a certain amount of time. After that, an assay called WST8 was used to analyze the survival rate of the cells after 24 hours, which led to the result that all species, including their precursors, showed near 100% viability rates, therefore fit for human consumption. The second and the third criteria would be the particle size and finally the long-term antioxidants. So the sampling methods for quantifying these values are as follows. We first stirred the moth solutions in a hydrochloric acid solution in which the pH is set to 2 for 2 hours. After that, we continuously stirred the moths in a buffer saline called phosphate buffer saline, PBS. This was to mimic the intake pathway of the moth once they are ingested, going through the stomach and the small intestine and then absorbed into the bloodstream. So every 12 hours, a small amount of sample was acquired and centrifugation was held. For the precipitated MOFs that were sunk beneath, TEM, transmission electron microscope, analysis was conducted to get the temporal change of the particle size distribution, which led to the result like this. So before we evaluate it, I want to bring up with a short explanation that in order for the MOF particle to have the maximum circulation time within our body, their particle size should be within the range bigger than 10 nanometers and smaller than 1 micrometer. In the case of SU101 and FETHQ MOFs, most particles were under the size of 1 nanometer, fulfilling the criteria. Where in the contrary, CuC81, many particles were over 1 micrometer. For the supernatant on the acquired samples, the concentration of the antioxidants present in the supernatants was analyzed using something called UV-Vis spectroscopy. Then their unit was converted into something called ORAC, a commonly used unit to deduce the amount of antioxidant properties of a chemical. This led to the result like this. First off, for SU101, there was near zero antioxidant production, as you can see on the y-axis scale. However, in the case of FETHQ, it exhibited over 5,000 ORACs worth of antioxidant production in the first 12-hour interval. After that, it still constantly produced antioxidants between the range of 400 to 800 ORACs. Finally, for CuC81, it showed better performance compared to the first one, SU101, but still had minute amount of expellation, less than 50 ORACs. This is a comprehensive result of the 3 MOFs, showing that FETHQ was selected as optimal in both particle size and long-term antioxidant effect. So for conclusion, this is a table that sums up the methods I presented earlier, showing that FETHQ showed optimal performance in all 3 criteria: particle size, biocompatibility, and long-term antioxidant. Now, I mentioned something called ORAC units, and it might not come close to you, so I'll just bring up a comparison. The amount of ORAC unit that FETHQ produces every 12 hours is equivalent to 21 times the antioxidant effect than the most potent blueberry that is known to contain antioxidants. For conclusion, FETHQ is a promising long-term antioxidant in the terms of being strong, yet consistent, also being biocompatible, and finally, having a synthesis method of being rapid and simple. This would be the end of my research, and thank you for listening.
Thank you. That was really interesting. So, what inspired you to research in chemistry and medicine?
What inspired me most was I read a book called The Secret Life of the Periodic Table, which introduces 118 different elements and their history and their applications throughout human history. So I got the idea that a minute change of electrons, the number of electrons and number of neutrons, could lead to having diverse chemical structures. And I could see the possibility that if I delved in as a chemist, then I could manipulate these into creating new materials that haven't existed before. So that's what inspired me to become a chemist.
Awesome. So when did your research journey begin, and how long did it take you to get the results we've seen?
The research began about May last year when I was in high school, and it took about a little over a year, so yeah.
Awesome. So when looking at these MOFs, have you considered the possibility of other kinds of reactions in the body that might potentially be harmful?
So the thing about the biocompatibility and a biology-related experiment that we conducted in this research was all in vitro, so outside our body. So in order to get the complete mechanism and know what's actually happening when these MOFs are introduced in our body, in vivo analysis and clinical trials have to be conducted, but we haven't reached that phase yet.
Awesome. Thank you so much for your time.
Yeah, thank you so much.
Big round of applause for Woojin Park. The next project is about minimizing the electrodes needed for an ECG to allow faster treatment for the patient. I want you to give a warm hand for the guy from Germany, Bastian Auer.
Hello, my name is Bastian Auer. I'm from Germany and I'd like to talk about my project ECG channel reconstruction using convolutional neural networks. First, something about me. Who am I? I'm from Germany and I came here during my research from Jugendforscht. It's a national competition for young scientists in Germany. I've been researching at Helmholtz Center in Germany. It's a public center for researchers in Germany. And that's where I came up with my idea. In my free time, I mainly do some kind of work with computers and with neural networks, and that's how I combined it with my job. As my job, I'm a paramedic in Germany, and then I came up with the idea to optimize ECGs because there are some kind of problems. The main problem at current ECGs is that you need 10 electrodes for the human to you need 10 electrodes for full diagnostic ECG. The problem with those electrodes is that there are 6 electrodes you need to place on the chest and 4 more you have to place on the limb of the patients. The big problem are the 6 ones on the chest, and I tried to reduce them. The problem about them is that it takes about 4 to 6 minutes to place them there, and they are very sensitive to movement, and it has a very slow diagnosis due to that. The even biggest problem is that you have to place them on a very specific place at the patient. If you only have a margin about 2 centimeters, if you are off like 6 centimeters, it won't work anymore. The even worst thing about it is that it takes so long. And then I tried to do something about it that it will get faster. My idea was to reduce all those electrodes needed from 10 to 4 to reduce the burden for the patient and for the emergency team. How did I do that? I used neural networks and put the data of 4 electrodes into them. What are the benefits of only 4 electrodes? They are easy to apply. It doesn't matter if you apply them at the bottom of your arm or at the top of your arm. They can be easy applied. It only takes about 10 seconds to apply them. The diagnosis is possible within a few seconds. And what's also possible, there's a one-patch design where you only need 1 patch you can apply to the human body without any cords or anything like that. And what's also good about it is that it's very cost efficient. What's good about it is that it's not that sensitive to movement as the usual 6 electrodes. The 6 electrodes, you aren't able to move anymore, you aren't even able to speak, because if you would so, the recording of the ECG will probably won't work probably. And with only 4 electrodes, you're able to move, and you are even allowed to speak. What are my results? You can see them right there. I put all those 4 electrodes into a neural network and combined them using CNNs. I told the neural network as an input, use those 4 electrodes, resulting in 6 channels, and output 1 of the missing lead from the chest. Those are my results. You can see the orange bar on the bottom is the average error of my neural network training. It means that the loss is very low because it's nearly 0. But what you can also see in this diagram is that there are some high errors up on the top shown in red. How does that happen? I talked to you that those electrodes on the human body, on the chest, are very sensitive to movement. That's what happened here. The original data set shown in green and the reconstruction shown in magenta are very difficult, very different. The green one, probably the patient moved or some kind of electrode got loose and you wouldn't be able to diagnose anything from it. On the other hand, the magenta one works perfectly fine and you can diagnose properly from it. This shows that even the reconstructed ECG works better than the original one. These are also my results. You can see them right there. That's one 12-channel ECG you would get if you go to a doctor. On top, the three lines are the original ones we put on the body, the leads on the limb, and the one V1 to V6 are the ones the neural network reconstructed. At the bottom, you can see them laid on top of each other. The green one is the original one. The yellow one is the the magenta one is the reconstructed one. You can nearly see that they are nearly the same, and they lay ideal on top of each other. That was my project. Thank you for listening.
Very inspiring. So, how did you get interested in research and ended up submitting this to the contest?
I didn't really get interested into research. I just did it, and then I was here. That's how it went.
Are you considering doing more research in the future?
Yeah, I'm probably going to do more research in the future. I'm currently in a small company, and we are doing a lot of research.
So the ECG results we see, where did you get them from? Was there some database, or did you test on the person?
There's an online database called PTBXL, and you can get it online with 20,000 ECGs, and that's where I trained my AI from.
Cool. Do you see this being applied to your own job as a paramedic in the near future?
I hope so, that we will be using it in the near future.
Do you have any advice for people who see a problem and want to try solve it?
Just try and solve it. If anyone tells you that it doesn't work, just try it.
Awesome. Thank you so much.
Thank you.
Big up for Bastian. Thank you. The next presentation is about unlocking insights into environmental flow programs, success and challenges, promoting the sustainable water management. From China, Xiaoyan, Diana, Dai.
Thank you. Hi guys, my name is Diana. I'm from the University of Melbourne in Australia, where actually it took me 21 hours flight here, so it's like really a long way. I'm gonna talking about improving the effectiveness and efficiency of monitoring programs of environmental flows. So the first question is, what is environmental flow? So under the current climate change challenges and biodiversity decline trends, our rivers are increasingly important. Environmental flows, also known as e-flow, representing water releasing from a dam or weir to the river systems to make our rivers happy and our environment healthy. However, the river network are just so complex, including a lot of aquatic species like fish, microinvertebrates, and riparian vegetation, and some outside world like rainfall, wind, and temperature. So the questions about like how much water do we need, and of what time of the year should we put water into the river, and at what part of the river should we put water in are really critical. And all those allocation decisions need careful planning, and here comes to the effectiveness monitoring of e-flow programs. So my research mainly focused on the human aspects that we believe the values and attitude of the people who working in those programs can make us to make the program better. And our ultimate aim is to make the program of higher effectiveness and efficiency. So moving on to my methods, I am using one notable example as a case study known as flow monitoring, evaluation, and research, in short, FLOMER projects across the Murray-Darling Basin in Australia, as you can see from the figure here. The Murray-Darling Basin is the largest water catchment in Australia. It covers the southeast of Australia and southeast of Australia, and covers around like 1 million square kilometers, just about the size of like Spain and France joined together. So what I have been doing is I have conducted 16 interviews for the people who are working in this program, and based on the interview results, built a more general survey, and there are a total number of 60 valid responses. I just want to mention you guys, like this FLOMER programs have two main components. The first one is the monitoring and targeted research at 7 selected areas, and the basin scale evaluation where they actually took the monitoring data of the selected areas. And this program has been run for 8 years already to understand the effects of water delivering to the river to the ecological systems. So the results I'm going to present today are two simple questions. What are the strongest benefits and the most pressing concerns of this program? Here is a list of strengths I identified during the interview and surveys. So from top to the bottom, I arranged them in terms of the rank, but I just want to be clear that the bottom one, contribute to academia, is not an undesirable characteristic. It is just the criteria not coming to people's mind when they think about the good aspects of the program. So just let's take a look from top to the bottom. We have monitoring of multiple objectives over time, improve system understanding, and consistency of core measurements over time. So I just want to include a direct quote from one of my survey's respondents saying, this program is world leading in terms of scale and complexity and of global significance. But what about something that we are like not willing to see? So here are the identified pressing concerns, and top one represents the most pressing concerns, like similar to the others. While having these two figures aside each other, we can find something interesting. Like for example, while monitoring for multiple objectives are a clearly highlight of the programs, people are saying the objectives are vague. So there are two types of objectives in this program. We have basin scale and selected area scale objectives. But there is a clear strength that the objectives exist across different spatial scale, but they are often defined in comparison to a pre-environmental water scenario, which we don't really want to see. And also, sometimes the objectives are not clear enough. That makes our assessment difficult. Like for example, it is really hard to quantify the success of improved fish abundancy without specifying a focal species and its targeted biomass or density. Another thing I found really interesting is the inadequate spatial coverage. Like that's totally the most pressing concerns we need to address for this problem. So what we can think of, there's two reasons. So the first one is, even though the monitoring is conducted at selected area scales, those monitoring is not adequately enough for even that portion of the basin. So this is a common design challenge for the monitoring programs that combine multi-catchments or multiple sub-areas. Another thing we can think of is the monitoring sites might not fully representative for the selected areas, as they might not fully capture the characteristics of the variability of the riverine networks. Even though the monitoring sites were carefully chosen, the site access and familiarity were major considerations. So what I've been presenting today are really simple, and I just want to give a really brief idea about what those projects look like and how important they are. But besides that, I also want to, mentioned that like integrating human perspectives and decision making into the technical science are really important because it is an interdisciplinary approach that helps us to understand the complex interplay between human activities and our natural world. And this interdisciplinary approach will help us to develop some strategies for the natural world and environmental challenges. And that's it from me today. Thank you very much.
Thank you. That was very interesting. So, what made you interested in studying environmental flow?
Yes, so like environmental flows is a type of restoration projects that because of we're building dams, so we're in tops of rivers, so the flows we got at the downstream are not as much as the natural flows we got without those infrastructures. But those infrastructures are just important because we need water for our crops. So environmental flow is just an idea how we can restore our rivers to the natural flows, and I think this is a very important concept under the changing world, and that's like my research topics is mainly around this one. Yeah.
Very interesting. What were some challenges you encountered during this research?
Yeah, it was because this research is mainly like talking to people. So approaching them and talk to them in a nice way is really important because the people I mean like working with, including the like, leaders of these big projects, they are actually the people who make decisions about like millions of dollars, like where the money goes every year. And I don't want to like upside them, like say in front of them, like this is the bad of your projects. I just want to say, oh, maybe this is the way that you could do things better. So like communication and in a really nice and polite way is kind of difficult. I don't want, yes.
Did you offer constructive tips to them?
No, I would just say, yeah, just like it's, yeah, sorry.
No, I understand. It makes sense. So do you think research like this can help enable better projects for observing environmental flow in the future?
Yes, I do. So in terms of environmental flows, there are a bunch of like modeling and statistical analysis that help us make decision better. But I think like talking to people, and because Australian has a pretty strong indigenous background, so we do care about what people say and what local community accept the idea or not. So just talking to them and let them accept the idea will just help both them and us, and the delivering of the management intervention better.
Yes, so very interesting. And did you encounter any insights when interviewing people from different parts of the project?
Yeah, actually it is. I was like, so before we are interviewing managers and scientists, and we thought their opinion could be different. Like managers might be more care about how things go, and they care about if this is successful or not, while scientists might more care about their scientific merits, but actually we turn out find, oh, their mindset are pretty similar, and this is kind of one of the unexpected findings we got from this research, so quite interesting, yeah.
That is very interesting. Thank you so much.
Thank you.
Big applause for Diana. And now the last presentation out of 4. This research focuses on discovering new previously undiscovered cancer DNA mutations. From the US, Kevin Xu.
Hi everyone, my name is Kevin Zhu. I'm a current first-year university student at Stanford University, and I'm sponsored by the Society for Science and the Public from the United States, or ISEF, to present here at SIYSS. So, in the last 50 years, global cancer morbidity and mortality rate have rapidly increased, with over 30 million annual cancer cases expected by 2030. Now, given that, one of the most effective predictors for cancer survival rate is the stage at which cancer is diagnosed. Now, in order to increase early cancer diagnoses, we need to be able to understand how cancer forms and identify new genetic mutation markers in cancer to study. we know that 2/3 of all types of mutations in cancer are known as replication errors, essentially DNA that replicates. And a subset of this, known as tandem repeats, have recently become valuable to study. Take, for example, a sequence of DNA like CAG repeating 20 times, so CAG, 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 CAG 20 times. What I'm looking for is essentially changes in that repeat size. So instead of 20 repeats, maybe it's 200 repeats or 2,000, or all the way down to 5 or 0 repeats. And so these types of mutations are not easy to discover in cancer due to the high genomic variability or instability in cancer, which causes sequence ambiguity. Essentially, the genome sequence itself, the DNA, may be inaccurate, hard to read, or the algorithms we use aren't as useful as or accurate as they could be. And so kind of how we get around that is we utilize current algorithms and improve upon them. And so my innovation was I collaborated with the group at Illumina to create a catalog called the Active STRs catalog, where we basically chose regions in the genome that have a high likelihood or probability to show different lengths or changes and mutability within the genome itself. Over 170,000 locations were included in that catalog, over 60,000 of which are non-reference based, meaning that these appear essentially out of nowhere in the tumor samples, and so we know where to look for them. The other thing I did was develop something known as local read depth normalization, essentially accounting for false positives by normalizing the accuracy or the read depth measurement in the genome sequence. And so kind of why this is important is I'm looking at genetic biomarkers in cell-free DNA. So after we're able to identify that mutations exist in cancer, how do they function outside of just those tumor cells? Could they be found in the bloodstream where the DNA shears off of the tumor masses and ends up in the bloodstream? And can they be used as localized measurements or diagnostic measures? And so the very first thing I did was I utilized the dataset called a pan-cancer analysis of whole genomes, which contains over 2,500 matching tumor and normal genome samples from patients across 37 different cancer types. I then used algorithms, the ones I created as well as existing ones, along with the catalog or prioritized locations in cancer to look for these mutations. And what I found was that there was over 182 instances where within these cancer patients, we found significant differences in the lengths of those DNA sequences repeating across 10 different cancer types. Now what's really interesting about this figure is that the large majority, shown in the blue, of these mutations only occur in one cancer type. So for example, mutation A only occurs in liver cancer, or mutation B only occurs in kidney cancer. That means that if we're able to identify that this mutation occurs in a particular patient, we can tell them not only do you have cancer, but you also have just kidney cancer or just liver cancer. So the diagnostic value here is extremely important. And so from there, I took a case study looking at 8 of those mutations in liver cancer. And then within 454 patient samples, I looked at the lengths of those repetitive sequences of DNA only at those 8 locations and used a simple regression machine learning model to basically ask, okay, does the lengths of these locations have any association with whether an input sample is cancerous or not? And I found that at nearly a 98% accuracy rate, we're able to tell, okay, this sample is tumor or not. And so from here, I wanted to work with actual patients and validate that these actually exist within patients and not just on a computer program. And so I worked with patients within the lab that I was working at and gained access to a tumor tissue sample, a normal tissue sample, and a blood tissue sample. And for each one, I extracted DNA, sequenced the DNA, and looked for changes within those regions to see if that repeat size actually changed. And you can see here, right, in the normal sample, so the normal person's DNA, that CGC repetitive DNA sequence is found. Whereas in a tumor sample at that region, that contraction occurs, so that sequence is not there, showing that in actual patients, this mutation does occur. From there, I wanted to test to see if those mutations would end up in the bloodstream. And so I gained access to the blood samples or the plasma samples of those same patients and tried to see if that DNA from the tumor would travel into the bloodstream by looking at primers. And I found here, you can see these 4 instances where that light essentially represents a mutation being found within the blood samples of those same patients. And kind of what the conclusion here really is that this catalog that we've used for the first time has over 174,000 locations for us to look for these potential mutations. And that these mutations, called tandem repeats, are a novel type of mutation that we've discovered that can be categorized more and may have functional roles in cancer to study. We also know that within liver cancer, on the previous slide with the case study, that they're actually usable as a diagnostic measure. In terms of future investigations, there are several things that we can do. One is to investigate the presence of them outside of just liver cancer, and then develop different ways for us to look at new mutations. And lastly, correlate them to molecular settings. For example, understanding them in cytotoxic activity or relations to cell death. And I kind of want to just end the presentation by going over kind of the application here. If you were to walk into a clinic one day and take a blood draw and have that blood sent into a laboratory where the laboratory can then extract the DNA, process the DNA, and then conduct genome sequencing on it, a computer at the laboratory with an algorithm that I created could essentially tell you, okay, you have predisposition or a likelihood of having liver cancer or kidney cancer just with a blood test. And so it eliminates the barrier of having to do a surgical procedure or a biopsy to figure out whether you have a particular cancer type, saving costs and increasing safety for patients who may not have the ability to have those types of surgeries. And second is allows us to discover cancer much earlier on if they were to present with this kind of DNA in the bloodstream rather than a tumor or symptoms showing. Thank you, and I'm happy to answer any questions.
Very cool research. So the first question here is, why short tandem repeats? What drew you to investigating them?
Yeah, that's a really good question. To me, short tandem repeats was kind of like a math problem, right? You're looking at repetitive sequences in DNA, and like if you take a string, right, it's just like the same thing again and again and again. And at the time when I started doing research, it was almost 3 years ago now, I was trying to learn how to program. And so to me, I used it as an excuse to build my programming skills and see how I could actually identify these types of regions. And then kind of from there, I ended up discovering a lab that was working on them and applying the concepts I learned into real-world scenarios.
So kind of jumping off from what you just said, your research has a few facets, like there's the data analysis aspect, and there's also the live using patient tissue. Which part was most fun to you?
Oh, I would say two big things were really fun. One was on the computational end, programming, right? Just kind of seeing the results out there. The most like, I guess, rewarding part of programming is that end result, and it can be very fast, a very fast pace. Whereas on the patient end, you're actually talking to these patients, understanding what their life is like, and telling them that we're trying our best to help other people in your situation. So kind of those two aspects were the biggest thing for me.
Awesome. And so you kind of went into this a bit when you were talking about future research to be done, but do you have any guesses as to what factors affect the number of repeats in terms of gene?
Yeah. There's definitely a bunch of different pathways that we can think about, but one big thing is telomeres or repetitive sequences of DNA, right? And cancer is uncontrolled cell growth. So there is some sort of association with the number of repetitive sequences we see and the potential for the formation of uncontrolled cell growth. So maybe there's a relationship there with the telomeres, or it could just be gene expression, right? These tandem repeats originally were associated with neurological diseases, so Huntington's disease, et cetera, et cetera. Those pathways are known to have effects with genes and function of expression. So I'm sure that there's some association with these tandem repeats in their locations, whether it's an exon or an intron, and how they code for proteins and so on and so forth.
Yeah, that does make sense. Do you have advice for aspiring young researchers who want to get into work before university?
Yeah, I mean, I think it's very daunting at first to go through the process, but the research I did was simply through a cold email. Like, all I did was read a paper, do some stuff on my own, and send an email out to professors that were in the field, and I gave value to them, and they gave value to me, and from there, it came together. So try your best and go for it, even if it seems like it's a long shot.
Fantastic. Thank you so much.
Of course. Thank you.
Thank you, Kevin. So inspiring. I hope some of you are inspired to participate in the exhibition arranged by Federation of Young Scientists in the beginning of next year, or this spring, right?
Yeah, this spring you can submit your high school diploma shortly, I believe. I'm not sure if it opened just yet, but it will within a few days or weeks. So shortly you can submit your high school thesis to be a part of the exhibition.
Thank you to Elin, thank you to Armand, thank you to everyone organizing this fantastic day. And a big round of applause for our participants. Thank you.
And before we finish off this SIYSS Day, I of course want to thank you, Julia. Here is a goodie bag to show some of our great appreciations for the work and job you've done here today. Big round of applause to Julia.